Okay, so I'm going to go ahead. I'm going to start with the PowerPoint. I'm recording. I'm going to hide these floating meeting controls. So today we are going to try to find biological. This is the wrong one. Oh, no, it might just have the wrong title. Yeah, I have the wrong title here. Differential expression analysis. There we go. Okay, so to put this in perspective, where have we come from? We started out talking about basic biology and bioinformatics. We learned how genomic information is passed on starting in the DNA. It is transcribed into the RNA. We call that expression. The mature RNA is called mRNA, and that's what we measure when we're doing RNA sequencing analysis. It's transcriptomics, it's transcription. And then that's translated into the protein. 3 bases at a time are converted into a different language, the language of amino acids, and those amino acid strands get built into a protein. Then on our second lecture, we learned about basic genomics databases. Where can we go to find information about the DNA sequence of humans, of mice, of pigs, of any of these species that we like to study? And how can we learn about the gene information? We have our favorite gene. Can we get the sequence for that gene? Can we see where the proteins are made in that gene? Then our third lecture, we introduced next generation sequencing. How can we take a sample of biological tissue, we put it on that sequencing machine, and it reads base by base our mRNA sequences. And if you remember seeing that little video, we saw flashes of light depending on what that strand of RNA's bases were. And it kept track of tens of thousands of different strands of RNA getting sequenced at the same time. And that's the parallel aspect of the sequencing. And it made so much of those short reads. And that's the high throughput aspect. And we learned how we could take a bunch of different samples, tack on little barcodes, and pool them together so that after we sequence, we can separate those barcodes, those samples out, and have separate sequences. Then in our fourth lecture, we learned about NGS databases. We learned about the gene expression omnibus, where you can search for publicly available expression or ChIP-seq data sets that are similar to what you want to show with your hypothesis. We learned about TCGA, which is the Cancer Genome Atlas, where they have compiled hundreds of samples from a bunch of different types of cancer and provided RNA-seq, proteomics, DNA, metabolomics, methylation, copy number information, just all of these different types of measures on all of these different types of cancers. We learned how to download this information. We learned how to do some easy visualizations on their websites. So that brings us to today, where we are going to demonstrate how to download that TCGA data and do differential expression analysis. Then next week, we're going to do functional enrichment analysis, what a lot of people like to call pathway analysis. So let's get into this. Today in our lecture, we're going to be talking about importing, manipulating data from TCGA and R. We are going to perform the differential expression analysis. It's going to do statistics. And then we're going to look at different ways of visualizing those results. That is our differential expression pipeline. So remember back, I guess 2 lectures ago, we were talking about counting reads. And I want you to remember that when we talk about a count is representing a single sequenced read mapping to a gene. And most of these fall into our easy to count category because it is very clear this red line read belongs to gene A. And when we talk about the depth of coverage of gene A, it has to do with at any point in this gene, how many reads are stacking up in gene A. There are other ways that the reads can map to the gene. It's because genes are complicated and we can't really count those in our count. Now, another thing we need to be aware about when having this RNA sequencing data, you should be thinking about this when you're planning your project, is how many replicates do you need? We need technical replicates sometimes, but we need biological replicates all the time because people are different from each other. If you're doing a human disease project, you're probably going to be taking samples from human tissue. And everyone is a little bit different. And the signals of measurements that come from each individual going to be a little bit different. And that difference may or may not be due to the disease. It just might be due to these being different people. Now, if you're doing a mouse experiment, it is assumed that these mice, they are inbred so much that the actual underlying DNA sequence of one mouse is the identical DNA sequence to another mouse. I don't know if that's exactly true. There are differences, mouse to mouse to mouse, but there are going to be fewer differences. There's going to be less biological variation between mice than there will be between people. But no matter where this data is coming from, we need replicates because there's going to be a lot of uninteresting variation in the data. So we need enough consistent measures that we can make out what is the interesting variation, probably caused by disease or leading to disease, than the uninteresting person to person differences. The goal of our differential expression analysis is to determine for one gene at a time whether the differences in expression between our two main groups is different enough given the variations within our replicates. Sometimes we also might have technical replicates. A technical replicate is checking the sequencing technology. So we take one sample and we sequence it twice, the exact same underlying sample, but sequenced maybe twice on the same machine or on two different machines. And that would be testing the technical sequencing differences. And that is useful information because sequencing is not always exact. There are errors in what the sequencing machine does, and sometimes we need to see those errors or understand how much those errors are contributing. Okay. Another thing we have to think about is statistics. So we are doing a statistical test by comparing Group A to Group B. And anytime we do a statistical test, we need to choose a distribution or a modeling method that where that distribution's assumptions match up the best with the data that we have. Now, reading about some of these distributions, you would read that there's a binomial distribution, and this is used a lot of the times with counts data. So thinking about the probability of getting so many heads when you're flipping a coin. That is counting, just very simple counting. There's another distribution called the Poisson distribution, and this is used in situations also dealing with counting, but when the number of events are very large and the probability that what you want to happen is going to be very small. So a lot of events are happening, but the probability that you're going to get the winning lottery ticket is very small. So we would model that type of distribution with a Poisson approach. So thinking about our RNA sequencing data, we have counts. So these are counts distributions. And we have a very large number of genes. We will have 30,000 to 60,000 different measures. And the probability that one or a few of our genes is our disease causing gene is very small. So, looking at this, it might make sense that a Poisson distribution could be appropriate in this situation. But if you read a little bit more about the Poisson distribution, you would see that that distribution assumes that the average is equal in magnitude with the variance. So as the average goes up, the variance goes up the same amount. But we see in RNA-seq that genes with high expression also have a higher variation than genes with lower expression. We'll see the slope on the next slide. So the people who decide statistics and pick the optimal distribution to model this difference have decided that we should use the negative binomial distribution because it allows for instances where the variance is gets bigger faster than the mean does. For the binomial distribution, the variance is less than the mean. For the Poisson distribution, they are equal. And then for the negative binomial distribution, the variance is greater than the mean. So that is why the statistical test that is used is based on a negative binomial distribution. This is a way that a tutorial online chose to demonstrate this. They had counts data, they measured the average and the variance of their counts data and plotted it. So on the x-axis, we have the average. For the y-axis, we have the variance. So each dot here is a gene, and it's showing the relationship between the average of that gene and the variance. So how much variability does that gene have? And if everything was equal, you would expect it to follow this red line. So this is average equals variance line. And you'll see that in this instance, the variance increases more than the average. So this falls above that unity line. So this is demonstrating how RNA-seq data, the variance is larger than the mean. And it explains why we pick the negative binomial distribution for our statistics in RNA sequencing. We don't need to know how to do this by hand. It's all done within the differential expression package, but it's good to understand what the package is doing. Okay, so that builds us up to be able to understand what we're doing. But first, we're going to download our data set. So these were the commands that I used a year or two ago to download the counts table, the probe map information, mapping ensemble gene names to gene symbols, and the phenotype files. And I've changed some of this. Like I don't do this step where I parse out the sample name. You did something similar to this in the homework, but I realize I don't have to do that. So ignore that part. But we do, the data we download from Xena, it is not exactly counts. It is log2 transformed counts. And so we need to unlog transform that. So I'll show how I did this in the code later, but we do that first, and then I filter the counts. And this is the part that I did for you before you got the data, just to get sort of the computationally heavy part out of the way to share smaller files with you. But let me go to TCGA just to remind everyone how you would get this data. Xena browser. I just always search for Xena. It already knows that I go there. So you search for Xena, UCSC, and it will make sure you don't get Xena Warrior Princess. So make sure it's the TCGA browser. Okay, and then which one we're doing? We have LUSC. So every cancer in TCGA has a 4-letter code. Some of them only have 3 letters, but it's a code that's unique to every cancer. And I got LUSC because that's the first one I clicked on. I choose the expression and I choose counts. There are other options here. There's FPKM, upper quartile, and there's TPM. And these are 3 different normalized expression shares. And I'm going to talk about this normalized expression in a couple of slides. But for differential expression, we need this counts table. And let's see, this is what you would click on to download the counts table. It's gzipped, but R can import that. It's fine. This is the gene mapping table. You just click on that. It will download. This unit is important because it tells us that these counts, they've been log transformed. So that's how I know we need to unlog them. The other thing from TCGA you need to get is the phenotype data. This is also gzipped, but it's fine. You can import this directly into R. This gives you a preview of what it looks like. It doesn't have all the columns here. There's 90 different columns, but this is our phenotype table. I've already downloaded it. So the TCGA data that I have downloaded here, it's from the previous analysis release. So the phenotype file should be the same, but this probe map file is unique to this counts table. If you were to go to TCGA, this website, and download this again, you would need to download this probe map specific for this star counts table. So always download them together, keep them together. I did check, I downloaded this star version and this probe map version, and without changing the script too much, I was able to analyze this newer data set. So it's the same samples, basically the same information at the end, but it's a different analysis process. So it's going to give slightly different results. So that's how to get your data from Xena. For differential expression analysis, we're going to use DESeq2. It comes from Bioconductor. And what it is, it assumes that most genes are not contributing to the difference between groups. And it looks at the variance in your data and shrinks it because genes that are changing should be changing a lot, and genes that are not changing, it's going to try to minimize the influence of those genes. If you want to read the vignette or install the package, here are links for those. This is the process that we're going to go through in our script later on today, but DESeq2, it has a function for putting your data in the DESeq2 object. For that function, you're going to need to provide it with a counts table. We already have it in all counts only. We have our sample table fed into call data. And the very important thing for this counts table and sample table is they need to have the samples in the same order. The sample table needs to have samples as rows, and the counts table needs to have samples as columns. And it will check, are your column names equal to the sample names in the samples table? And then the third thing we need to specify is what is the column from our sample table that we are using to compare. This column, sample underscore type dot samples, has the tumor normal information. And that's what we're comparing here. We are comparing tumor samples expression with normal samples expression. And this first command puts these three things into the DESeq2 object. Then the second command will estimate size factors. I'm going to talk about this, I think, in the come right here. Estimating size factors is the way of normalizing the counts data because we need to correct for sample to sample library size differences. And this means the number of reads for sample 1 compared to the number of reads for sample 2. We need to correct out those number of reads differences. And I'll talk more about that, but that's what estimate size factors does. Normalizing our sample differences. The third function, estimate dispersions. This is setting us up for our statistical test. It's going to measure the biological variance from gene to gene. So how much change is this gene having between our groups? And it shrinks them. It's trying to minimize just general gene expression changes that are not biologically relevant. They're just minor changes throughout our data set. They're not strong, they're not meaningful, and so it's shrinking the variance of most of our genes. And then this n binomial walled test. This is our negative binomial statistical test. It's going to fit this negative binomial and apply walled statistics to get us that p-value out of it. And we'll talk about p-value a little bit later. But that is our statistical test. Negative binomial walled test. And then results simply pulls the results out of our DESeq2 object. So up till now, this DDS1 is a sort of a complicated DESeq2 object. But then we pull out the results, save it in a table. This results function will pull out the average expression across all of our samples, the log2 fold changes, so how much is this gene changing between our groups? Standard errors, test statistics, p-values, and adjusted p-values. And in a couple of slides, I'm going to explain why adjusted p-values are important. We can use the summary function to get a really quick summary of our results object, telling it cut our p-value off at our adjusted p-value, off at 0.05 and give us the summary. So it's telling us 31,000 genes were tested. There was a cutoff of an adjusted p-value less than 0.05. There were 41% of our genes, they had a significantly upregulated change. So they had a positive fold change. They were higher in our first group. And then 25% of the genes were downregulated, so that they were more highly expressed in our second group. And this first and second group is very important to set. Otherwise, it's going to be alphabetical. We set the groupings with a factor. We factor our column, we set the levels so that our primary group is like our disease group, and our secondary group is whatever we want that control to be. With a tumor normal setup, we want the tumor to be our primary group and the normal to be our secondary group, our control group. And if we didn't set the levels of our factor, it would be switched around because normal comes before tumor in the alphabet. So be sure that you're always setting your factors to what you want them to be. If you want to simplify your code, you can replace these 3 lines of code with just this DESeq function, and it will go through, normalize your counts, shrink your dispersions, and do your statistical tests all in one go. Okay, so we've already talked about what we need for differential expression. We need sample groups with replicates. I would say at a minimum, you need 3 replicates per group. I will not do differential expression for anyone without 3 replicates. With only 2 replicates, I tell them I will calculate fold changes, but no statistical tests. So make sure you have at least 3 replicates in each group. You need your counts table. Make sure they are counts. A good rule of thumb is if these numbers are whole numbers, if they don't have any decimal places, it's probably counts. But always go back and check that source. Make sure what the website or your collaborators are providing you with are counts. Because all of DESeq2's assumptions and methods, they are based on counts. And if you give it normalized data, those assumptions are invalid. Then the third thing is your sample information. Sometimes this is called metadata. That's the cool new thing to call it. I call them phenotypes. Also see them called clinical data. So this is descriptions of your samples. You really only need one column of this. You need to know what are your groups. Sometimes it's useful to know more information. More information is always good, right? So think about what's available, or if you're designing your own experiment, what can you measure? What can you describe in your sample information that would be useful to illustrate in your data set? So so we can talk about these inputs a little more detail. Where are you going to get your counts data? Accounts matrix. HTC count is a program that can generate 1 file per sample. You could use one of those new RNAseq abundance measure methods like Salmon or Callisto that skips the alignment and just does counts. So your counts could come from there. Always make sure that the rows of your samples equal the columns of your counts and that the samples are in the same order. The design should be what column from your sample you want to compare. You can also make a more complicated design. So in our example, we're just comparing a group of tumor samples with a group of normal samples. But if you know anything about cancer and how cancer is studied, is that this normal sample is coming from the same person that the tumor came from. So this is called a paired analysis because you have a tumor from your patient and you have a normal measure from your patient. So if you have that for all of your patients, you could set up this paired analysis where you tell the statistical test that this is the sample base column that I want you to pair these samples on. So it will take that into account when it does the statistical test and correct for that redundancy, knowing that this tumor, this normal, came from the same patient. You could also include covariates in this setup. Do you need to correct for sex or for age or for some important clinical measure that was measured with this data? So I do statistics for a PI that studies lung cancer. And when I do the statistical test, he says, I know these measures, they also need to be included in the statistical test because they are important. And so I'll include those in my setup here, include them as covariates. But just in the simplest format, you need one column with group information. That is all you need to compare. Okay, so I talked about that we need to normalize the sample to sample differences. And I'm going to demonstrate why. Oh, we have chat. Sorry. Is it different from getting data from Geo? Geo is complicated because each listing in Geo can share the data in slightly different ways. Like you look at the Geo listing and there's usually a link you can click to download the counts table. Some geo listings, they'll only share normalized counts, and those will not be good for doing your differential expression analysis. So look specifically for geo projects that have counts, and there will be a link to download the counts on that web page. It's usually down at the bottom with the supplementary information. But you also have to get the phenotype information, which if they make it easy, they'll share that as a sample table too. Okay, and then our second question is, what method are they using to adjust the p-values? This is going to be a false discovery rate correction. It's the Benjamin-Hochberg method. And I don't know if I describe in detail how that happens. But the quick basics of it is it sorts the p-values in order and does a calculation for each p-value to sort of make it a little bit larger, depending on what order that p-value is in the list and how many more p-values there are. It's a really easy thing to calculate in R. There is one function and you can adjust your p-values, but it's all taken care of within DESeq 2, and we don't really have to think about it. Okay, let me get, haha, okay, another question. For Geo, I found UMI counts versus read counts. UMI implies that there is a unique molecular identifier for each count. I would get probably just read counts. Make sure that this is not like a single cell data set. As long as it's a bulk RNA-seq data set, then probably just read counts. I don't know. Mhm. Okay. If you have a question about a particular geo listing, send me the link to it by email and I'll look at it and see what I can find. Like they're always just a little bit different and like they'll share the counts in different ways. But yeah, I can look at them. I've looked at lots of them. Yeah. Okay, so we are gonna have this thought thought process. So we have two samples here. One sample one, we've sequenced 100 reads. I know this is a rubbish sequencing experiment, but we have sequenced 100 reads for sample one and 100 reads for sample two. So it's completely even. And our gene A, we have 10 reads for sample one and 10 reads for sample two. Let's see, we have 100, sample one, sample two, 100 and 100. For our gene A, we have 10 reads in sample one and 10 reads in sample two. So the proportion of reads mapping to gene A in each sample is the same. So we can say sample one and sample two have the same expression level of gene A. For gene B, we have 10 reads in sample one, 20 reads in sample two. We can get our proportion here. I know this is a very silly example, but it's demonstrating something that I want to show. So for gene B, our expression level, we're getting 10% of our read counts in sample one and 20% of our read counts from sample two. So we can say sample two expresses gene B twice as much because twice as many reads are mapping to sample two. But this assumes that my trackpad is all messed up, so I can't click and move. Okay, this assumes that both samples have the same number of reads. If they have the same number of reads, sequence from the start, we can just compare read counts, and it makes sense. Twice as many read counts, twice as much of that gene. But what if, for some reason on our sequencing machine, sample two had twice as many reads sequenced? Sample one has 100 reads, sample two had 200 reads. We map our reads to our genome. We find that for gene A, sample one has 10 reads and sample two has 10 reads. So we might think these samples have the same expression level for our gene, but we have to think about what is our starting material. So for gene A and sample one, we're capturing 10% of the genes, where for gene A and sample two, we are just capturing 5% of the genes in sample two. So gene A in our second example, sample one has a 2 times expression level of sample two. If we look at sample B and look at the reads that map to sample B, we see gene B. We see sample one has 10 reads. Sample two has 20 reads. So looking at just the reads, we might think sample two has more expression of gene B. But we need to think about the number of reads that were generated for our sample. We use that information to figure out what is the proportion of our reads mapping to gene B. And we see 10% of our sample one reads are mapping to gene B, and 10% of our sample 2 reads are mapping to gene B. So gene B has equal expression between our 2 samples in this example because of that read difference between our 2 samples. So this is why we normalize the sample-to-sample differences. We look at the number of reads that were sequenced for each sample. This is called the depth, the depth of reads. It's also called the coverage. How well is that sample covered with reads? So you might see that terminology, depth or depth of coverage. That's just talking about how many reads were sequenced for that sample. And a lot of the time, so let's make this a real number. We have 50, 1, 2, 3. We have 50 million reads sequenced in sample 1. We have 100 million reads sequenced for sample 2. Our gene, let's make more realistic counts. And gene B, just random numbers I'm typing. Okay, so what is often done for a super simple correction method? We were just dividing. We divided the number of reads in gene A by the number of reads in sample 1. That gives us, if we just do that, it gives us some super small numbers here. That's not really a meaningful way to keep this measure. So what it's done is it's divided by the counts divided by 1 million. And this is called counts per million. When we divide the number of reads in our sample by million and use that as a correction factor to divide our gene count by. So that gives us sort of a number that we like to look at. 1, 2, 3. There we go. So this is norm gene A and norm gene B. These are expression values. These are our normalized expression values in counts. This is a count per million. This is our million. And with these normalized expression counts, we can calculate a fold change. So what is a fold change? It has directionality and it has a ratio. So let's say this is our tumor and this is our normal. We're going to calculate our fold change. So we want our tumor to be the highest value. We're going to take this divided by this. And we're going to take this divided by this. So this would be our fold change. This is not log transformed at all. What you're going to see out of differential expression is log transformed fold change. So it just looks better on plots. And you can, as you know that a value of 0 is a fold change of 1, and a value of 1 of a log fold change is a fold change of 2. Log 2 fold changes messed me up at first, but I kind of can think in log 2 fold changes now. Okay, I'll save this. Sample Diff expression. So here we normalized our expression and we calculated a fold change. What DSeq2 does to do this normalization, it It does it a little more sophisticatedly, but it's sort of the same idea. It calculates a scaling factor to normalize out the differences between samples. It does this with the median of ratios method, and I'll talk you through that method, I think, on the next slide. It's possible to normalize out the differences between genes of longer length or shorter length, but with differential expression, we really only look at one gene at a time, so it's not important to do that. But okay, but this is why you might want to do that. If you're going to compare gene A to gene B, then you would need to consider the length of the gene. So shorter genes, just because they're shorter, they have less room for reads. Whereas longer reads, they're going to get more reads because they're longer. And so to correct for this gene length difference, we divide by the kilobase length of the gene. This is where you get your FPKM, your RPKM. TPM is something similar as well. It's going to correct for the sample depth, so the differences between samples, and the gene length. FPKM, it stands for fragments, so our reads per kilobase, where the kilobase is the length of the gene per million reads for that sample. So the depth of that sample. And that's why it's called FPKM or RPKM, transcripts per million, similar to. So if you see those terms, know that's what it means. Okay, so what is DESeq2 doing for this scaling step? Estimate size factors. So it takes your, let's say you have 6 samples, and it calculates a pseudo sample, which is the geometric average of all of your samples, and that is your sample average. They call it a pseudo sample. Then it calculates the ratio of every single sample compared to your pseudo sample, calculates a scaling factor for that sample as the median ratio across those genes, and then divides the count for each gene in that sample by the scaling factor of that sample. And it's very similar to doing that counts per million correction that we just did by hand. But this is what DESeq2 is doing behind the scenes. So we talked about importing our objects into DESeq2. The first thing we're going to do with that data is we're going to apply a variance stabilizing transformation to it. And this is just an additional normalization on top of this normalization. It's going to log2 transform it and stabilize some of the variance in that data. And it's this VST normalized data that we're going to put through a principal components analysis. So we know we have a few samples, at least 6. We have a bunch of genes. We have 30,000 to 60,000 genes. That is high dimensional. It's a lot of measures. It's hard to get an idea of the big picture of that type of data set. So we apply a technique called principal components analysis, which is going to take those 30,000 measures and collapse them into just a few measures. This is a dimension reduction method because it's taking all of our measures and collapsing them into just 2 in this case. It provides a view when we plot these 2 measures of which can describe these two measures. They describe most of the variation in our data set. And we can take these two measures and plot them, shade it by the samples information that we have, and get a view of our data. This allows us to understand whether the differences in our data can be explained by any of our clinical measures. It's very good if this difference in our data, so you can see there's two clusters, can these two clusters be explained by the groupings that we want to use for our differential expression analysis? Are there any samples that are not clustering with their group? So in this one, there was one sample that clustered with, it's a tumor sample that's clustering with the normal samples. And I saw that and I figured that is probably a tumor sample that has been misclassified. It's not really a tumor sample. It's probably a normal sample, and it was just given the wrong name in the tube or something. There's something going on here, and we should probably remove that sample. Something else that we can see from this data, so we're shading here based on the tumor normal assignments. And we can see these normal samples, they're a lot like each other because they're clustering very tightly. They are very similar in expression profile. Where the tumor samples, there's a lot of variance in them. The catchphrase for the James Cancer Center is no two cancers are alike or every cancer is unique. I should always look this up because I can never remember it. But there's a catchphrase for the James Cancer Center that we are demonstrating right here that all of the cancers, all of the tumor samples, they have a lot of differences. If we compare this spread of tumors versus this spread of normal samples, we can see that. Okay, so that's the first thing we can look at with our data. Before we even do the differential expression analysis, we ask, what do our samples look like with a PCA plot from that principal components analysis? So we do our differential expression with that command, and with statistics, you always see a p-value. And the p-value is telling us what is the probability of getting the results that we are seeing right here. We set a cutoff for our p-value to state what percentage of the time are we okay with being wrong. So we set it at 5%, and we say when we do this test for one gene, we're okay getting it wrong 5% of the time. But when we're doing this test over and over and over again, 30,000 times, that amplifies. So 5% of these 30,000 genes, they're going to appear to show a difference, but that's just a random wrong test. What we found, big difference, is just wrong 5% of the time. So we apply a multiple testing correction to make sure that the change we're seeing is consistent enough to be a true difference. We use this FDR method, a false discovery rate, called the Benjamini-Hochberg method. There's a bunch of different methods, but this method multiplies each ranked p-value by the total number of genes and divides by the rank. So it sort of minimizes the smallness. It makes the p-values very slightly bigger. And if your p-values were small enough, then some of those adjusted p-values will also be small enough to pass your adjusted p-value cutoff. That is my quick and dirty description of a p-value and an adjusted p-value. Sometimes in DESeq2, you'll see p-values set to NA, and that's its own method of filtering the results. So if a row count has an extreme value, like most of the counts, they're pretty average, but then one or two samples, they have an extremely big value. Then DESeq2 will flag those and say, even though there seems to be a big difference in expression between our two groups, it's because some outliers are there in our data. So we're going to set the p-value to NA and you can ignore those genes. So that's why that happens specifically in DESeq2. So after we get our differential expression analysis, we have some ways to visualize it. The first way we talk about is with a volcano plot. This is a way of visualizing the relationship between fold change on our x-axis and p-value on our y-axis. The p-value is negative log 10 of that p-value. It's usually the FDR value, but we just the name says p. But this means that very small p-values have a very large number because we are logging them. So the larger this number, the more significant our gene. So every dot here is a gene. And large negative log 10p, very important, very meaningful. Then on the x-axis, this fold change, the bigger the fold change or the more extreme negative fold change, those are going to be our most interesting genes. Big difference between our groups. A positive fold change means it's higher in our test group. A negative log fold change will mean it's higher in our control group. So you can say these red dots, these red genes, on this positive side with a very small adjusted p-value, those are your most interesting on your tumor side. On the other side, the negative side, these red dots, they are most interesting, more highly expressed in our normal samples. We use a package called Enhanced Volcano to plot this. There are some options. You can have gene symbols labeled or not. I'll show that. You can adjust the p-value cutoff. You can adjust those fold change cutoffs. There are so many options with Enhanced Volcano that's why I use this package. We can also pull out our most interesting genes, so maybe all of these red genes, and plot them on a heat map. For heat maps, I like to use a package called Complex Heat Maps. We take the expression of these genes and the normalized expression, and we scale that normalized expression. So for one gene, we are going to scale that expression so that average is centered around 0 and the standard deviation of this gene is set to a +2 -2 level. So we are scaling expression. If this gene has above average expression, it's going to be shaded here in red. If this gene has a below average expression, it's going to be shaded in blue. And we scale these genes because different genes are going to be on different scale levels. So gene A might have a really high expression level, where gene B might have a medium expression level. And we could plot those, but coloring the plot based on expression, it would not be meaningful because when you put them all together, it doesn't look good. It doesn't tell us the useful information. We can play around with this later and I can demonstrate it. But anytime you make a heat map, it's going to look more informative if we scale it. The other thing we do in a heat map is we cluster our samples. So samples are columns here, so that samples that have an expression profile for our selected genes, they are put closer together. We also sample our genes across the rows so that gene A has a similar profile of gene B. They're going to be put closer together. Then one more thing I want to point out on this heat map is we can provide sample annotation. We can provide gene annotation. It would be on the rows here, but you can set up annotation for whatever you want to describe for your samples. Here, we are coloring this bar based on whether each sample is a tumor or a normal. And this gene set, it's very good. This gene set we've picked is very good at distinguishing our primary tumors in this first group in green against our normal samples in gray over here. So that is a check for your heat map. Are the genes you picked out good at clustering your samples into the proper groups? Another thing I like to check out just to make sure I factored my groups properly is the direction of fold change going in the direction I want it to. So the first gene had a very positive fold change, and we see it's higher in our tumors, where a gene with a very negative fold change is expressing higher in my normals. And this is my check to make sure I factored those groups correctly. Okay, so we're going to run through this hands-on activity in R. Think about, after we do this, what would you do with this information? We're going to get a list of genes that are different between our two groups. How could you use that to guide your future research into lung cancer? So, okay, I'm not even going to talk about that. Okay, so if you want to follow along, be sure to download this zip. Hopefully you've already done it. We'll go through this. We've talked about a lot of the functions already, but if I don't get through all of this today, we only have 15 minutes. I will finish it next Tuesday when we talk about functional enrichment analysis. Okay, because we only have 15 minutes. So I set up the script with the packages. If you need to install any of these packages, then you can uncomment the line, install your package, and comment it back. Because here are the packages we need to use. I have this date here. I save it as P, just so I could see how long it takes to run through the whole script. It takes 2 minutes if you highlight everything and run it. So, but we don't have to do that. Okay. Next thing we're going to do is set our working directory. It's here in the hands-on folder. I'm going to paste that in here to source. Okay, it's saved here. I'm going to save that. So this first chunk, I have it commented out because we don't have to run it. I am giving you filtered process counts, but on my own time, I downloaded this HTC counts tsv.gz file. I imported it into R. I did this unlogging. I took the logged counts. I did 2 to the power of that. That unlogs something. And then I subtracted 1. and then I rounded it to have 0 decimal places to make real counts. Then I did a filter. I wanted the total counts across all of my samples to be greater than or equal to 2,000. These are going to be the genes I'm going to keep. If there are at least 2,000 counts in that row, I keep those rows. This is an index that's going to tell R which rows to keep. Then I subset my table based on those index rows. Then I save this table into our filtered counts text file. And that's what we have here, filter counts. That's what you have. And you can go straight to reading that file in. It's still a pretty big file. It has 28,000 rows. The rows are genes. The columns are samples. There are 550 samples in this data set. And these are counts. They're whole numbers. We also have a gene table. It's going to translate ensemble IDs to gene symbols. That's all we need from it. In our phenotype table, we have a bunch of clinical information. I don't even know what all of this is, but there are descriptions on the GDC TCGA website for every one of these columns. We will see that we have phenotype information for 765 measures, but only gene expression for 550 of them. So we need to process this to make sure our count samples are equal to our expression samples. First, we're going to take a look at the measure, the column from our sample table that we're going to use. We see there are primary tumor, we have solid tissue normal, we have FFPE scrolls. We are just going to keep the tumor and the normals. So we've gotten rid of those scrolls. And then I'm going to limit this sample size just to make this analysis go faster. We still have a bunch of samples, so I'm going to limit it to only those samples with a tumor stage of 2A. That drops us to 88. We have 65 tumor and 23 normal. I'm going to save this value, the number of phenotype columns, and the start of gene variable. I'm going to use these numbers later to know where to cut my table. And then I'm going to let me show you these sample IDs. I'm going to G-sub this dash for a dot, because if we look at our counts table, when R imports column names, if they have any dashes in them, they get converted to dots. The sample names in our phenotype table, they need to be processed to match that. So that's what we're doing with the G-sub. So that is our first processing. Now we're going to combine our counts table and our sample table for two reasons. We want to make sure they have exactly the same samples, and we want to make sure those samples are in exactly the same order. So first, we're going to transpose our counts table. We're flipping it to its side so that we have samples as rows and genes as columns. All it did. So that we can merge this table with this table, counts and phenotype, and we're going to merge them based on this row name. That's it, merge right here. The weird, annoying thing about merging tables is it takes your row names, puts them as a column. So we are throwing those back out to be our row names. Here, they're back out here. And we're going to remove this column. We do not need this capital R row names anymore. So we have merged our table, cleaned it up a little bit. You can see these first few columns, they're all phenotype information. If we scroll over, eventually, there's a lot of it. Eventually, we're going to hit genes. So we see the phenotype information, it ends at 125. The gene information, it starts at 126. That's why we made these variables, number of phenotype columns and start of gene. There's probably better ways you could do this, but this makes sense for me. We are going to pull out our counts from our table. They go from, we're using this square brackets notation to specify the first column we want to pull out at and the last column, which is going to be pretty much the rest of the table minus three columns because we do not need these last three columns. This might sound familiar. This no feature, this ambiguous, and this alignment not unique. These are those instances from my picture I keep showing over and over again. These are the counts that couldn't be assigned to genes. So this is kind of like junk counts. And we don't need that in our counts table. So that's why our end column, it's 3 minus the very end of our table. Go back 3. So it ends right here. So we get our all counts only. And then on the front end of our table, we're getting our phenotype information. It's going to start at column 1, and it's going to go to that 125 number we made earlier. So it's the first 125 columns of our table. We're going to take our sample group information. This is our tumor normal. And we're going to factor it. It's just a character right now. We're going to factor it and set the levels to be normal tumor. And this does it seems backwards to me. That's why I always have to check. It seems like we should put the tumor first and then the normal, but it does it the other way around. So now, let's see, go back to our table. Now it's been flipped back around. And I've just done that. I have some code in here if you're interested in doing this a slightly different way. I have code that will look for paired tumor normal pairs. So what it does, it looks for submitter ID. So this is that base sample name that exists 2 times in the data. And it filters based on that. So out of this commented code, you would get a table with tumors that also have normal samples. Because right now we have 7 normals and 65 tumors. So not all of these tumors have a paired normal. So we're just treating these as 2 different groups. If you want to try it, paired tumor normal, this is what you would run. And then you would run your differential expression with that sample group information. Okay, so probably what we have time for today is to talk about this PCA plot. So first, we take our counts and our samples. We put them in a DESeq format just to look at the PCA. We are not including any group information. Now, we could, and it wouldn't change anything for what we're trying to do here. But first, we just get a quick look at our data, we put it into our DESeq2 object by providing it with our counts table and our sample table. And then we take this and we normalize it with this VST function from DESeq2. It is quickly estimating dispersion and applying a stabilizing transformation to get a normalized expression table. It does something very similar to what we're going to do later with estimating size factors. But VST is for quickly getting a normalized expression table so that we could pull that out. Well, we don't need to pull it out quite yet. We're taking this VSD object. It's a DESeq transform object, and we're feeding that into our PCA to plot it. What this is doing is it's taking the normalized expression of all of these genes, collapsing it into two dimensions. PC1 is the dimension that explains the largest percentage of the variance, and then PC2 can explain a little bit more of the variance. And if you're calculating this outside of DESeq2, there's actually more PCs that it can calculate. You could have a three-dimensional PC plot. It would have PC1, PC2, and PC3. So there are more PCs. But for this example, we just care about PC1 and PC2, and we tell it what do we want to shade it based on. And our main interest is this tumor normal difference. If we had another, where's our phenotype table? We want all samples. If there was another column of clinical interest, let's see. Those only, we need something with a lot of data. Let's try this one. New table. Okay, there's some no's and yeses here. We could shade based on a different thing, different column from our sample table. Which one of these samples had a new tumor event after initial treatment? So I guess the blue ones here, they grew more tumors, where the green ones, they did not experience any more tumors. So this is really only interesting for this tumor side. So as you're thinking about your particular analysis for your differential expression, if you're pulling from TCGA, are you going to compare normal versus tumor, or are you going to compare something different? So something that might be interesting about the tumors. And if it's going to be that latter, think about maybe you want to limit it to only tumors first. Like, are you going to compare deceased versus alive? Limit it to only tumors and then compare deceased versus alive, because it's not going to make sense to have those normal samples clouding up your comparison. So that is my warning. Really think about what you need to focus on and filter as you need to up here. Limit it to only the tumor samples. Select, I don't know, tumor samples with a KRAS mutation versus tumor samples without a KRAS mutation. How is that KRAS mutation affecting the gene expression? It might be interesting. So we are going to stop this here. Next Tuesday, we'll come back, we'll catch back up with the code, and then do actual differential expression. Write those results to a file, plot our volcano plot and our heat map, and box plots. And then the actual new stuff for next week will be functional enrichment analysis. Okay. So that was differential expression in a nutshell. We're not quite done with it, but that's a good start. Okay. You can stop sharing. Stop recording.